Okay, I make that the hour, so I think we'll get started. Hello, good afternoon, good evening, or good morning, depending on where you're joining us from. And welcome to this Twist Bioscience webinar, Harnessing the Diversity of CRISPR-Cas Proteins for Genome Editing. My name is Chris Thorne, and I am Senior Manager for Field Marketing here at Twist. Okay, so it gives me great pleasure to introduce our speaker for today, Dr. Gedrius Gesounas. Dr. Gesounas is Chief Scientific Officer at Cas9 and also a Senior Researcher at the Institute of Biotechnology of Life Sciences Center at Vilnius University. He obtained his PhD in the laboratory of Professor Virginijus Šiknis in 2012, during which he studied the mechanism of Cas9 protein activity, demonstrating that it is the programmable RNA-guided DNA endonuclease. He is the co-author of 28 scientific publications, which are cited more than 3,000 times, and was involved in several Cas9-related patent applications. So, with that, Dr. Gesounas, I'd like to hand over to you, and I'll let you take it from here.
So, thank you. So, good afternoon, everyone. First of all, I would like to take a chance and thank Twist Bioscience for opportunity to present our work related to characterization of novel Cas proteins at their webinar. And as I was already presented, I work for a company called Cas9, and yes. Cas9 was established in 2017 and is located in Vilnius, capital of Lithuania. Main founder of company is Professor Virginijus Šiknis, who is known for contribution to elucidating of Cas9 molecular mechanism. So I was a PhD student at his lab at the time, and he have a chance also to contribute to this work. And also later, Dr. Monica Pauli also joined our team, and we decided to use our knowledge in CRISPR field and experience for commercialization, and that's how Cas9 was established. So as a company, we focus on three main activities. First one is to provide research services for CRISPR applications. Next, we explore new Cas proteins for various applications, and also developing and characterizing new CRISPR-based molecular tools. Our main competences are not limited to CRISPR biology, but also we are working with genome editing tools and applications, cloning expression, purification of proteins, and also in vitro protein and RNA characterization. Since our goal is to develop new tools for gene or genome editing, I will shortly present this technology. So to introduce desired change in genomic DNA, firstly, double-stranded DNA break should be introduced in specific loci of genomic DNA with using endonuclease, and then DNA repair machinery of the cells repair this double-strand break, and depending on selected strategy, various changes could be introduced into genomic DNA. And it's difficult to overestimate the importance of this technology, because in the research, I think researchers in many labs, researchers are using this technology for molecular studies in molecular biology. Also, it is important in medicine for treating genomic disorders and also some kinds of cancer. And importance also in medicine could be illustrated that started several clinical trials to treat diseases such beta thalassemia, and also it is important to consider to be a future of agri-sciences for advanced plant breeding, because to meet the challenges which humanity facing because of climate change and also expanding people population. Most popular tool, endonuclease, used for introduction of these double-stranded breaks is Cas9 endonuclease, which is sometimes compared to molecular scissors. And Cas9 protein binds to a small guide RNA, which helps to recognize specific DNA targets. So, but to bind the target, firstly, Cas9 should recognize short DNA sequence called PAM. Then one DNA strands are separated, and one DNA strand is duplexed with this guide RNA, while another strand is displaced. And the two active sites in Cas9 called RUF-C and 18H cleave opposite DNA strands, leaving double-strand DNA breaks, which are required for genome editing technology. So, in few years after elucidating after showing the function of Cas9, it was demonstrated that actually Cas9 could cleave the genomic DNA in various cell types, including human cells, animals, plants, and bacteria. And when double-strand break is introduced, it is repaired by two major cell pathways. One is called non-homologous end joining and is error-prone and leads to small deletions or insertions or simply indels, and usually leads to gene inactivation. Another pathway is homologous recombination. It requires also homologous templates with homologous arms be present in a cell, and it could help to introduce larger changes such as gene replacement or gene correction. And despite the wide use of Cas9 protein, this technology still have several limitations or drawbacks. First, since Cas9 target is dual, as I mentioned before, one part of the target is recognized by, called protospacer, is recognized by RNA, while another part called PAM is recognized directly by Cas9 proteins through interactions with amino acid residues. And PAM is characteristic to each different Cas9. So even though PAM sequence for most popular Cas9 protein from Streptococcus pyogenes or spike Cas9 is only NGG dinucleotide, it still has some limitations for applications like base editing, or if you want to edit 80 genomes like plants, or for targeting only single allele. Also, mismatches between guide RNA and target DNA sometimes are tolerated. Therefore, in addition to main target, Cas9 protein could cleave other targets within cells and introduce unwanted mutation. And this problem called of targets problem. And last but not least, spike Cas9 is large protein, about 1,400 amino acid in length, and it do not fit well into most popular viral vector, adeno-associated vector, for delivery in vivo, which is needed for disease curing strategies in adult organisms. From previous work before establishing Cas9, we knew that we characterized several Cas9 proteins and knew that different Cas9 proteins usually from different bacteria usually recognizes different PAM sequences, and also that different Cas9 proteins can have different specificity or lower number of targets, especially those enzymes which recognizes longer PAM. So to look at to expand this Cas9 portfolio, we decided to analyze Cas9 proteins which are present in sequenced bacterial genomes. And Cas9 proteins is part of so-called CRISPR-Cas systems, which confer adaptive immunity against bacteriophages in bacteria. And these CRISPR systems consist of CRISPR region, which is also composed of short DNA repeats, about 30 or 40 nucleotide length, which are interspaced by unique short DNA sequences called spacers. And when phage invade bacteria, two proteins from CRISPR Cas locus called Cas1 and Cas2 recognizes this foreign DNA and insert small fragment of the phage into CRISPR region as new spacer. Then this CRISPR region is transcribed and processed into small CRISPR-RNAs, which then is bound by effector complexes which degrades invading DNA sequence. And CRISPR-Cas systems are very diverse, and actually there is quite complicated nomenclature to name them all, but some nucleases from class 2 CRISPR systems actually requires only one protein for both guide RNA binding and also target cleavage. Therefore, they are very interesting for gene editing and other applications. And Cas9 proteins from type 2 CRISPR systems are most abundant, but also there's found another family of proteins called Cas12, which differs a lot by sequences and also by protein length, and that divided at least in 10 families. So initially we decided to focus on this most abundant Cas9 family and analyzed all Cas9 sequences available at the time. I think it was about 10,000 different Cas9 proteins from different bacteria species. And we built sequences also varied a lot. So we built phylogenetic tree and distinguished at least 10 different clades, and then selected more than 100 Cas9 proteins from different clades of the phylogenetic tree to cover all the diversity of Cas9s. Also, usual size of Cas9 proteins varies from 1,000 to 1,500 amino acids. So we selected also proteins different in length to cover all this diversity in length. So next we needed genes for Cas9 protein synthesized, and this is where we use services of Twist Bioscience. So basically we use two services. One is synthesis and cloning of Cas genes into expression vectors, and also a synthesis of guide RNA templates for production of these guide RNAs. And the reason we liked this is fast synthesis time and also very competitive price. So when we have genes cloned, then we should, to establish the cleavage activity in vitro of each Cas9, we need to decipher two components. One is sequence of guide RNA, which are different differs between Cas9 proteins. So it could be determined from sequence. So therefore we analyzed all potential guide RNA sequences and confirmed it experimentally. And a second component is PAM sequence. So far it could not be determined from protein sequence. Therefore we needed to determine it experimentally. And for that we used 7 nucleotide randomized library where each nucleotide was changed into either 4 in each potential position of PAM. So after the digestion of such library, we selected cleave sequences and by deep sequencing determined PAM for more than 80 Cas proteins, and they PAM sequences were very diverse, and there was like more than 50 novel combinations of PAM sequences. There was A-rich, G-rich, C-rich, and T-rich PAM present. But next we wanted to find a link between PAMs, this recognized PAM sequence, and Cas9 domain, which recognizes this PAM sequence. But so a group of Professor Czeslavas Venclovas from Vilnius University tried to analyze all the domains of PAM recognition domains of Cas9 proteins, and actually these sequences also were very different. So they managed to group these sequences in 4 major clusters. However, PAM sequences between these clusters also varied a lot. So therefore, more data are needed to link PAM and sequence with this protein part which recognizes this PAM. Next, we purified more than 30 Cas9 sequences and analyzed cleavage activity in vitro. So we performed cleavage reactions in different temperatures and observed that actually some of, even though they come from bacteria, that some proteins are thermophilic and can effectively cleave DNA at 60 degrees of Celsius. And also, as I told before, Cas9 protein cleaves DNA, both DNA strands by 2 active sites at the same position. So for spike Cas9, they usually leave blunt end, but one of the DNA strand is additionally chopped. So we analyzed cleavage patterns of all Cas9 proteins and observed that some proteins leaves blunt DNA ends, while others forms 1 or 2 nucleotide starter DNA ends. So summarizing Cas9 work, we show that Cas9 orthologs could be a rich resource for potential novel genome editing tools because they recognize more than 50 new PAM sequences. Also, some orthologs are active in broad temperature intervals, and also cleavage patterns varies between Cas9 orthologs. And this data was published in Nature Communication journal last year. Also, our Cas9 orthologs could be used for other applications. It could be exemplified by work of Cardia Bio company, which is based in San Diego. So Cardia Bio is developing CRISPR chip technology, which enables rapid detection of specific DNA sequences in unamplified genomic DNA. So this technology relies on graphene-based field effector transistor, and on the graphene surface, Cas9 molecules are immobilized, and when Cas9 protein binds to specific DNA sequences, this binding event modulates electric characteristics of transistor, and binding events then could be detected. So Cardia Bio wanted to improve their technology to achieve single nucleotide detection specificity, and as a model they selected single cell disease related single mutation of single nucleotide. And when we try to choose target of spike Cas9, PAM was actually present of several nucleotides away from mutation. And as I said before, Cas9 proteins could tolerate some mutations between guide RNA and target DNA. So therefore, in this case, spike Cas9 is not very good choice. But in our collection, we found MJ Cas9 protein, which actually PAM is disrupted by this sickle cell disease related mutation. And actually, when MJ Cas9 was used for detection, it could discriminate between different single nucleotide variants, and also this Cas9 helped to discriminate between homozygous and heterozygous DNA forms. So this work, together with this description of experiments with our Cas9, was also published in Nature Biochemical and Biomedical Engineering paper this year. We started with Cas9 proteins, but also we are constantly looking for new Cas nucleases to our collection of proteins. So that's how we found Cas-beta proteins, which differs from other known class 2 nucleases. And similarly as Cas12 proteins, this nuclease has tri-split roof C domain at C terminus of protein, but other part of protein there was no similar proteins found for N terminal of the protein. Also interesting, even though protein itself reminds Cas12ase. The Cas12 proteins, this architecture of CRISPR-Cas locus is more similar to Cas9's. To test whether this new Cas-beta protein could cleave double-stranded DNA, we cloned modified system in E. coli vector. So modifications included T7 promoter in front of Cas-beta protein, and also spacers against our randomized PAM library in CRISPR-A. And for screening of double-stranded DNA cleavage, we used positive selection assay I described shortly in Cas9 work. And for this E. coli crude lysate with expressed Cas-beta CRISPR system was mixed with SPAM, incubated with SPAM library, and after cleavage, adapters were ligated to cleavage ends, and this cleavage products were amplified by and enriched by PCR, and by optimizing expression and cleavage conditions, we found finally get this PCR products showing cleavage by 3 of Cas-beta proteins. And by deep sequencing of this PCR fragment, we show that actually all 3 Cas-beta proteins recognizes CCY PAM sequence and also cleave DNA in PAM distal part of the target. Next, we analyzed which of elements of this CRISPR-Cas system, Cas-beta CRISPR-Cas system is required for double-stranded DNA cleavage. For that, we use the same positive selection assay, and when we deleted all 3 additional genes present in this Cas-beta CRISPR-Cas locus, actually this PCR product disappeared, suggesting that some parts from here might need it. Some components from here might need it for double-stranded DNA cleavage. So when we reintroduced the short DNA sequence between Cas-beta and Cas1, this PCR product reappeared, confirming that actually some components in this sequence are required for cleavage, and this it is most. And when we analyzed the sequence, we found between 3 available Cas-beta systems, we found that there is some conserved double-stranded RNA structures present, like hairpins, termination hairpins, or repeat anti-repeat sequences, which are characteristic to trace RNA. So this has led to design potential guide RNAs, which differed by the length of trace RNA and CRISPR RNA, and also presence or absence of termination like hairpin. And only one of these designs, which extended 5' and supported cleavage. So we use this design for other experiments. So when we had purified all components, so Cas-beta and RNA, we looked whether Cas-beta protein actually looked whether Cas-beta proteins is RNA-guided for confirmation that Cas-beta is RNA-guided double-stranded nuclease. For that, we actually mixed purified Cas-beta proteins with different guide RNAs and performed cleavage of DNA substrates. And actually, cleavage occurred only when specific guide RNA was present, confirming that guide RNA is the only component required for double-stranded DNA cleavage. Also, we identified potential active site amino acid residues in RUC domain, and introduced mutations to alanine, and this mutation abolished DNA cleavage, but not binding, confirming that actually RUC domain is responsible for double-stranded DNA cleavage, similar to Cas12 proteins. Next, we analyzed cleavage pattern of these proteins. So for that, we incubated the purified Cas-beta protein with this randomized PAM library, and then deep sequenced both ends of cleavage products. And actually, deep sequencing confirms that Cas-beta proteins leave 5' cleave DNA at PAM distal part of the target, and leave 5' protruding 5 to 9 nucleotide overhangs. And also Sanger runoff sequencing also confirmed similar results. Also, most of Cas12 proteins has auxiliary single-strand DNase activity. So when Cas12 binds to the and cleaves target, RUF-C active site stays in active form and can cleave any single-stranded DNA. And we screen for this activity of Cas-beta protein using single-strand M13-phage single-stranded DNA. And we found actually that when single-stranded DNA or double-stranded DNA, which is complementary to guide RNA, were present, there was degradation of M13 DNA. So it confirms that actually this Cas-beta protein, similar to type 5 enzyme, has this auxiliary single-strand DNase activity. Next, we aim to demonstrate that actually Cas-beta protein could be programmed to cleave any DNA sequence. For that, we selected 4 DNA targets in human DNA and observed and tested cleavage using linear DNA substrates in vitro. And surprisingly, at tested conditions, only 1 of 4 tested target was cleaved. So we selected two targets. One I will name good target, and another one bad target which was not cleaved, and analyzed this further. So when we increased temperature by 10 degrees, one of the proteins, Cas-beta 3, started to cleave this bad target, confirming that Cas-beta protein, at least some of Cas-beta proteins could be activated by temperature. Also, we compared cleavage of supercoiled and linear DNA substrates. So usually Cas nucleases, which is guided by RNA, cleaves supercoiled DNA more efficiently because in supercoiled DNA strands are untwisted and lower energy is required for strand separation. So in this case, both tested Cas-beta proteins cleaved supercoiled DNA more efficiently for both targets compared to linear DNA form. So these experiments confirm that actually temperature and supercoiling facilitates cleavage by Cas-beta proteins. And last but not least, we aim to look whether Cas-beta proteins could support DNA cleavage in heterologous cellular environment. And for that, we choose E. coli cells. And initially, those cells were transformed with plasmid encoding Cas-beta and specific guide RNA. And then strains already containing Cas-beta protein were transformed with plasmid which contains correct PAM and protospacer or lack the target. And after transformation, such cells were selected on antibiotic which resistance is conferred by transformed plasmid. And in case of Cas-beta 2 protein, we obtain transformants only in case when plasmid without target was used. So this confirms that at least Cas-beta 2 protein protects E. coli from exogenous DNA and could work in exogenous environments, so in different organisms. So summarizing Cas-beta work, so we show that despite sequence and differences, Cas-beta protein has similar biochemical properties as other Cas12 enzymes, and most probably belongs to type 5 family. So, summarizing my talk, I would like to say that also Cas9 not only perform R&D, but also offers great opportunities for potential exploration of Cas9 orthologs, other types of Cas proteins, CRISPR-based molecular tools, and novel gene editing techniques. And we believe that developing new molecular tools based on CRISPR-Cas technology could benefit to many therapeutic areas. So, in the end, I would like to thank all people from Cas9 who were involved in this work, and also our main collaborators from Corteva and New England Bell Labs, which were involved in all these projects I described in my presentation. So thank you for listening to me, and I'm ready to answer your questions.
Good, thank you so much. That was fascinating. It's honestly spectacular to see how this field continues to evolve at the speed it does, even now. So I guess if it's okay, I'd like to begin with a question that I have, which is, quite early on when CRISPR-Cas9 from S Biologies was developed, we also saw papers coming out that were using directed evolution approaches to improve that enzyme itself. So having identified all of these orthologs now, do you think there's room for improvement around those orthologs? Would you anticipate wanting to hone in on one or two of those for improvements, or would you rather expand the toolbox further, do you think?
Well, I think most probably combined approach would work the best. So I think maybe a good choice would be to just try to improve some most interesting enzymes with desired properties.
Mm-hmm. And what would be the properties that you'd want to prioritize, do you think, if you had to pick and choose?
Yeah, so as I mentioned in presentation, I think there's still several drawbacks of technology, but most important, I think, is that enzyme should have robust activity on eukaryotic cells without off targets, and also short pair. I presented a lot of enzymes, but most of them recognize as longer PAM, so shorter PAM also would be the preference.
Yes. All right. Well, we've got a few questions coming in from our audience. So if you do have a question, please put that in the question box. So I'm just going to attack a few of those now. So our first question came in from Bin Zhao from Stanford, who asked, how does the Cas-beta outperform other Cas proteins? So you mentioned there's various limitations and variables, but specifically for Cas-beta, how is it outperforming other Cas proteins?
So I think they have, as I demonstrated, still wild-type proteins cleave not all DNA targets. On linear DNA, it needs either temperature or supercoiling, and it is, I think, also characteristic to many other Cas12 proteins. But as some examples showed, it could be also improved by mutagenesis for other Cas12s.
Okay. A question from Emily Anderson at Horizon Discovery. Hi, Emily. So she asks, have you looked at the ability of Cas-beta to bind and unwind DNA in a deactivated form? So a form where you showed, in fact, where the catalytic domain has been mutated.
So yes, actually, we tested binding abilities, and this inactivated proteins bound target specific, similar to wild-type proteins.
Okay. A question from Jarrett Mann, who asks, with respect to a specific application, so can Cas-beta be used in place of a type 2 restriction enzyme in Golden Gate assembly? And I don't know whether that's something you feel you've been able to address.
Yes. So I think most probably Cas-beta as a protein could be used because it specifically recognizes this DNA sequence, what is required for cloning. But still, you should look for this sequence requirement to be sure if it fits well.
So a question from Balaz, which is, I guess, just looking for some guidance on where to dig into this more. So where can they find a detailed description of this positive selection PCR with adapter? I apologize if you missed that when you explained it. Is that going to be in the publication, or where can they go and find out more information?
So you can find this information described in detail in this Nature Communication paper on Cas9 orthologs. So I can send the link after seminar if you leave your credentials.
Sure thing. And then a question from Christoph. What kind of methods would you recommend for QC of on-target mutagenesis of the CRISPR-Cas?
Yes. So for if I understand correctly, question is what methods can be used for detection of on-target cleavage in eukaryotic cells. So we use at Cas9 we use either T7 endonuclease assay, which allows just quick evaluation of activity, but for more detailed analysis we use deep sequencing of this edited locus.
Okay. Well, perhaps one final question then from me, which is along the lines of what advice would you have for scientists working in their labs looking to access these different types of Cas9 enzymes? You know, where can they go to learn more and potentially choose the right enzyme for them, or educate themselves on how to work with these different enzymes?
So I think if someone is interested about this Cas9 variants, so you can find this information in last year Nature Communication paper where they are described in detail, and also you can contact me and I will be happy to answer it and help.
Fantastic. Well, Gedros, thank you so much for a fascinating talk. If there are any additional questions, please don't hesitate to contact us at Twist, and we can forward them on, or you can find Dr. Gasinas at Cas9 as well. Please do take a moment to complete our short survey afterwards, just to let us know how you felt the webinar went, and we look forward to seeing you at our next webinar in the not too distant future. So thank you once again, And all that remains is for me to wish everyone a great rest of day. Thank you so much.
Thank you.